But of course, all these events, the whole Alumni Association, and especially Coffee with EMBL, which I think was a great inventive step and example of the EMBL can-do spirit and just making things happen. But none of these things could happen without the support of the administration at EMBL. We're very grateful to that. And we're especially grateful to a very young member of the EMBL staff who has mentored and supported us throughout this journey. And therefore, we'd like to invite young Matthias Hense to come on screen. Matthias is much younger than me, as I'm sure will again point out to the audience. But we are so grateful for all the support that Matthias has done. And so we wanted to say thank you with a little Christmas spirit. And the best type is a nice bottle of one of my personal favorites. And Thomas, I think, also introduced this. So there's a bottle behind you, Matthias, of Buna Haben.
Not just only a bottle. What a surprise. Thank you.
We hope you'll enjoy it.
Thank you very much. I mean, this is Christmas spirit in the true sense of the word. I'm shocked. Thank you.
You see, you have two Santa Claus, not even one, two.
Yeah.
I'm not sure how young I will feel after consuming all of this, but I'll try to take it in small sips and truly enjoy it. That's fantastic. Thank you so much, and not just for this, but for everything that you and many others who have contributed to the Coffees with EMBL and the interviews, as you showed, have done throughout the year. As I've said on occasions before, but I say it again, for me, the whole journey has been really a joy. So thank you so much.
Great. And huge thank you on behalf of the whole community who take part in these Coffee with EMBL and the Alumni Association. Thanks for everything, Matthias. You'll be a friend for life, and we look forward to engaging with you many times in future. So thank you once again for all of your support and your help. Thank you.
Likewise, and once again, thank you. I wish we had 3 Nooshs and the 2 others could now appear behind you and offer something similar. So great surprise. Thank you so much.
Great. And with that with that said, I think it's time to start our discussion today with a fantastic range of guests who can tell us about the EMBL of the current time and the recent decade. And with that, over to you, Thomas.
Yeah, so thank you, Angus. So we restart our journey, and it is time to talk about the 2010. So one of the main things that happened throughout that time was the establishment of the latest EMBL site, so EMBL Barcelona. And we have here with us James Sharpe, the current head of the EMBL Barcelona. So hi, James.
Hi there.
Thank you for joining. So it would be great to perhaps start on a little bit of history and understanding how, the EMBL Barcelona site came about, and, what was the history and the beginning of that idea.
Thank you. Well, it's wonderful to be here again. In fact, I remember, I think one of the much earlier Coffee with EMBLs back during the COVID pandemic, and it's great that it's still continuing. So yes, EMBL Barcelona. We are the youngest site. We're actually 7 years old now, which is sort of actually funny for me to realize this, because we've always been saying we're the new site, we're the new site, we're the new site, and I guess gradually you become less new, but we're certainly newer than the other sites. And in terms of history, I mean, I suppose the short version of events is that EMBL as an organization, of course, interacts with many countries and is quite an outward-looking organization. And since quite a long time ago, there was a connection to Barcelona. That was actually from around about the time when I came to Barcelona. I didn't come to Barcelona for EMBL. I came to Barcelona in 2006 to join an institute called the CRG, which is which back then was a very new institute here and became a very impressive and world-level molecular biology genomics institute. And in fact, it had a lot of EMBL influence. It had a lot of EMBL alumni. And at the time when I came, also Luis Serrano, who was co-chair of one of the units in Heidelberg, joined the CRG. I had not had any connection with EMBL, but it was then that a partnership was created, partnership between the CRG and EMBL. It was a partnership on systems biology, which is what I was working on, and Luis. And that partnership then went on for 11 years. And during that time, the discussions then arose that the local environment was very sort of conducive and actually in many ways very similar to EMBL. So the CRG Institute, this is, I think, one of the big legacies of EMBL over these 50 years, as we've now seen, is the way that it's inspired other institutes how to organize themselves. And the CRG very, very explicitly modeled itself on EMBL. So it has the 5+4 contracts for group leaders. So group leaders have to leave after 9 years. It has a graduate program that's very much based on EMBL. It has a lot of its other rules and regulations are based on EMBL. Possibly this is one of the reasons that CRG became very, very successful. And it also meant that the territory was fertile here for thinking about going to the next step. And the next step, from EMBL's perspective, was not just to have a partnership here, but to actually set a unit here, a new EMBL unit here. So that started in 2017, and I was recruited from CRG to EMBL to head the unit. So that happened 7 years ago.
So cool. That's wonderful. And I actually remember that around my time, there was a big colony of Spaniards that throughout the time moved to Barcelona. And you were referring to some of them that I know. By the way, actually, a lot of Drosophilists. I'm a Drosophilist, so I follow a little bit of that genesis. So,
well, one of the ones that was in your clip just there of Fatima Gabawa.
Yeah, exactly. Fatima among the others. Yeah.
But honestly, I was trying to count the other day, and this is don't take this too literally because it was very back of the envelope. But the number of group leaders at the CRG that have been EMBL alumni is, I think, more than 15.
Yeah. No. But I mean, I think it makes your point of how the EMBL is sort of seeding in the places where the scientists are moving, and there's a lot of fruits, right? CRG is one fruit, but an EMBL site could be another fruit, and we have heard these from other sites in discussions with, for instance, Nadia Rosenthal about the EMBL Monterotondo. Another curiosity that I had, because we know that EMBL sites have different types of focuses. Nadia was discussing at length, the EMBL mouse biology that is done in Rome. So perhaps I wanted to ask you whether you can tell us about what is the specialty of the EMBL Barcelona, and what is the science that is coming out from there, or will come out of there, since the site is fairly new.
Absolutely. I'm very happy to tell you about it, of course. So the title of the sort of subtitle for the unit is Tissue Biology and Disease Modeling. And so, in fact, maybe I could show just a couple of images to help discuss what we see as tissue biology. So let me just share the screen here. So hopefully you can see that.
Yeah.
Excellent. So just to help a little bit this discussion, of course, EMBL, and very clearly within the name is the phrase molecular biology. Now, the focus for us is a little bit the idea that, what biology and a lot of science has been over decades and decades is trying to understand, what complex systems are built of, what bodies or organs and things are built of. And so you sort of go down in this direction until you get to the fundamental units, which are genes and molecules. But we also, of course, want to understand how these things all work together. So our emphasis is a bit more on the other direction, which is putting things back together again. And particularly out of these multiple scales, because biology is a multi-scale system, we're interested in this scale here. Not so much whole organisms, which is maybe a bit too much for us, but going beyond molecules and cells to tissues. And these are just 3 images that are quite sort of attractive of different things that are essentially tissues. One is an embryo, this here, but these 2 here are blood vessels. And a very important element is about trying to study things in vitro. So when I said in the previous slides, we're at a very exciting time, the exciting element that I'm referring to here is this wonderful sort of discovery over the last 10, 15 years of how much we can recreate of tissues in vitro, in the lab. Cell culture has been around for a very long time, but now 3D tissue models are happening in vitro, in the lab. And of course, one of the things that is sort of providing a bit of a revolution in is that in terms of model systems, you mentioned Drosophila earlier, one of the absolute classic model systems, and mice have been used as a model system for mammals. But funnily enough, if you get to the point of being able to build interesting, complex tissues, multicellular tissues in a dish, you can actually start using the human as the model system. So by going from in vivo to in vitro, you go from mouse to human, and then by going to human, it means that you can start to do the second part of our title, which is really disease modeling, but disease modeling using human tissues. And, in a way, our motto is biology is a system that builds. Maybe we can learn by building. So some of the stuff that we try to build is not necessarily for useful medical purposes. It's just building for the sake of understanding cells. And this has got pictures of the faculty, but it's got more pictures of these tissues. So tissues in a dish. And these are embryonic tissues. These are vascular tissues, sort of vascular networks, where the amazing thing about these 2 groups is that they can flow fluids, cells, fluids, and even blood through the vasculature in a dish in order to recreate the vasculature. And then we have organoids, and then we have computer modeling. So I'm also very proud that the unit is very diverse, both at a gender level and where people are coming from, and also a sort of expertise level. So a lot of the group leaders here were not originally trained as biologists, and this is also a hallmark, I think, of modern biology, is to bring physicists, engineers, and mathematicians in for a very interdisciplinary approach. So I'll stop sharing there.
So that's great. I mean, it was very nice to see the focus, but also it's great to see images. I mean, these are very beautiful images, and actually gives me the chance to remind people that this year we had a Cafatos lecture that is actually very, very close to the themes that you have discussed. In fact, we had Hans Clevers, that is one of the researchers involved also in these types of research, especially on the organoid side. But I wanted to ask, I mean, I think, one advantage, you said it yourself, is you can use the human being, or you can use human cells, and this clearly has an advantage because you can sort of model some aspects of disease better. I think the other advantage with using cells in a dish is actually that you can, provide also the aspect of the engineering. I mean, to shape the cells and to add functionalities in order to test perhaps a concept of a disease or a drug or a device. So are you also, involved into the bioengineering aspect of biology?
Absolutely. So engineering is a key kind of word for us, and in multiple different ways. So one direction is maybe the one that you're referring to in bioengineering. So bioengineering tends to be seen as sort of trying to engineer maybe something complex that is, let's say, useful. And a number of the projects here fall into that category. I'll give an example of one because it's very just recently come out. But actually, we sort of also use engineering in 2 other meanings. One is sort of engineering as a sort of a philosophical framework to understand how biology works or how it's evolved. So not us engineering things, but seeing organisms as like evolutionary engineering solutions to problems. And then the other one is maybe the sort of non-living side. I mean, we are very much into building and engineering tools to do our research. So one of the things that EMBL Barcelona has done since we started 7 years ago is we've managed to set up here in the building, the same building where the CRG is, we've set up what's called the Microfab Lab, which is the microfabrication laboratory. And with 3D printers, with laser cutters, with PDMS, with microfluidics, that's another type of engineering that is essential to us. So we engineer tools, we sort of engineer tissues, and we also try to interpret biology as an example of engineering from a philosophical point of view. So, and then this is all about, you know, this thing of putting things back together again, right? To say that the organism is, you know, in that classic phrase, more than the sum of its parts. You can study all the genes, you can study all the proteins, but we are not just a collection of genes and proteins. There are emergent behaviors that happen when you put cells together that you cannot see in individual cells, you know, like a pattern, a spatial pattern across a tissue. And even blood vessels, you know, a single endothelial cell in a dish, you can't tell if it's actually functioning properly. A functioning endothelial cell has to provide mechanical integrity to actually conduct blood flow. So you can only, you know, you could see that the transcriptome of an endothelial cell in a dish looks just like one in a body, but that's not going to tell you actually whether it's really acting as an endothelial cell. So putting it all together into complex tissues is for us a very exciting and important step.
Yeah, so it's a vessel on a chip, I guess. And I mean, you can do all sorts of stuff, right? I mean, I guess this is also for you know, for disease. And I mean, I guess you were mentioning in our discussion before an example with malaria and things that are very important for human health, I guess.
Yes, so this is very exciting. The paper actually just came out a few weeks ago in Nature, and it's a collaboration between one of our groups, Maria Bernabeuer's lab, with 3 other groups. It's a sort of international consortium. But what her lab was able to provide in this context of tissue biology and disease modeling is that they have worked out how to essentially recreate the human pathology of cerebral malaria in the lab. She was working on this in her postdoc in Seattle, and I was very, very happy and excited to be able to recruit her as a group leader here. She's originally from Barcelona and was originally working with malaria, but in Seattle got into this engineering fusion. And what they're essentially able to do is use human brain microendothelial cells, use human cell lines for parasites and astrocytes that help to make the blood-brain barrier, and then to flow human blood through the vessels that are created by a kind of stereolithography technique, and human blood cells that are either infected or not with the human-specific malarial parasite. Now, going back to the comparison with animal models again, there are mouse models of malaria, but the human malaria parasite does not infect any other species other than humans.
Okay.
If you do the mouse model, you're not recreating the human malaria disease. If you do it with human cells in a lab, you're literally recreating the entire pathology of what happens. It's a very sad, you know, disease, which is that affects mostly children, and the lethal element of malaria is a swelling and an inflammation at the base of the brain from the blood vessels there. They are able to recreate it, and in this study that just came out, they were able to actually use it to validate 2 antibodies that had actually been identified from some resistant patients in Africa. 2 antibodies that are effective at actually blocking this inflammation. And so although those antibodies themselves cannot directly be turned into therapies, they are a great stepping stone towards therapies for malaria, verified in human tissue, but in the lab. So it's really a wonderful example of the whole excitement of this thing. Really.
Yeah. So what I think is also great is the beyond of course, the importance for human disease is also the multidisciplinarity that is entails, right? There's a lot of angles. I mean, there's the technical angles, the medical angles, the biology angles. I guess also the ability of modeling, you know, a system that now you understand and you have on a dish somehow. So to move a little bit, you know, on a larger perspective, since you know, the site is rather new, and I guess you also had COVID in between, so things are probably slowed down a bit because of COVID. I wanted to ask you about the future. I mean, how do you see the site developing? What's gonna be, you know, super cool, or what you think is gonna be super cool in the science that the EMBL Barcelona at this point will do in 10 years?
Mm-hmm. So it actually brings neatly onto the other element that we are very keen on and focus on here. So, which is mathematical modeling, so computational modeling of multicellular systems. So this is about using computers, and it's very much physicists and mathematicians, but I would like to stress that this is not what we classically call bioinformatics. It's not about looking at, you know, sequence analysis and protein structure. It's about building actual dynamical models of biological multicellular systems. Now, I think, so we are very much pushing this and focusing on this anyway at the moment. My lab and one of the other labs, two of the labs here are primarily computational modeling. Many of the other labs are also involved in modeling. So we've really tried to emphasize this across the whole unit, and it very much feeds into, you know, the message that I'm trying to get across about the sum being great, the whole being greater than the sum of its parts, which is emergent behaviors, emergent things that happen when many cells are together that you don't see in individual cells. So to understand emergent properties, you very often do want to build sort of rigorous mathematical models, because otherwise you can't understand where these emergent behaviors came from, like oscillations or dynamical waves or these kind of things. So in terms of the future, essentially, it's to get more and more sort of realistic and sophisticated examples of these tissues in vitro, married together closely with the mathematical modeling. The mathematical modeling is what allows us to believe that we actually understand it. It goes beyond descriptive biology into explanatory biology. And honestly, in a way, the sky's the limit. I mean, specific things that are very obvious directions to go in is to bring in more of the immune system. Other ones are to bring in more of the microbiome, for example, in gut organoids. So the fact that these multicellular in vitro complex tissues represent, in a sense, communities or populations of different cell types interacting. And there are so many interesting and important processes that are about the non-trivial interaction of different cell types. The immune system being, you know, a great example. And since we've already got these systems with blood, it's a sort of a natural next step. And the microbiome, for example, studying it not in a tube, but in a living human tissue. So those are, you know, two of the kind of directions that we feel that the field will move.
Wow. So that is really fascinating. Thank you, James, for discussing this with us. I think it's also great because we have started touching on a few points with your beautiful images and with the discussion of you know, the data and the computational part and the modeling part. You know, a number of things that we'll discuss during today's meeting and event. So thank you, James. I'll give it back to Angus to continue. Thank you so much.
Thank you.
Thank you very much, Thomas, and thanks a lot, James, for giving us that insight into the fabulous work that you're doing in Barcelona. I think you raised a number of issues that are very germane to today's discussion, but also to what we've learned in our survey and discussions with alumni and staff throughout the history of the EMBL, of the really important role of the EMBL organization in pioneering technology development and building new tools and technologies that allow us to discover novel insights about biology, but also importantly, once those discoveries are made and the technologies developed, playing a pivotal role in actually disseminating the technology and training people throughout Europe and beyond in how to take best use of these technologies in their own research. And our next speaker today, our next guest, is Stephanie Alexander, who's going to be able to tell us about a super exciting recent development at EMBL in the field of imaging. And we saw the beautiful images that James shared a few moments ago. But Stephanie, please tell us then about the imaging center that EMBL built.
Yeah, so thank you for inviting me that I can take part in this great event. So yeah, maybe a few words indeed about how the imaging center, or how we call it the IC, really came about. And I think this idea actually originates even before I started at EMBL. So I'm here since 2014. But it originated, the idea, in 2010 or the early 2010s with two parallel initiatives, one of the cell biology and biophysics unit here in EMBL Heidelberg, as well as the structural and computational biology unit, where it was clear that both units saw the need that the latest home-built light microscopes developed here, as well as the cryo-electron microscopy workflows, should also be provided to researchers outside of EMBL. And this was not possible, or not, there was not the capacity to do this in the research units themselves, and also our core facilities did not have the capacity to take this additionally on board. So during the last EMBL program, or in preparation to the last EMBL program, both ideas merged, and it was clear, okay, we actually need a new externally facing service facility. The whole project basically started in 2016 with a lot of people involved in the fundraising to make it also possible that we have a new dedicated building that really is made for imaging and can house the latest technologies. So construction started in 2018. And then in the middle of 2021, we actually could move in with the team, with the instruments, the first instruments, and start to provide service. And yeah,
I think you've been quite modest in saying, yeah, fundraising had to happen, but obviously that was a big deal, because I'm sure that the amount of money involved was quite substantial. I don't know if you can share just a little bit more about, I mean, how were you able to support this?
Yeah, so as I said, it was many people involved. It was the coordinator, Jan Ellenberg, but also Matthias, whom we just met, was involved in a lot of fundraising. Ian Mattei, the previous director, administrative director, Christian Scherf. So it was really a EMBL initiative, and as we usually have to do when we want to build infrastructure, we need to talk to governments and the host site governments especially. So a lot of political fundraising happened in Berlin, as well as in our local ministry of Baden-Württemberg. And alone these two entities provided over 30 million euros to fund the project, to get the construction going together. And then we had also generous support by Heidelberg Materials. So we needed a lot of concrete, and this was made possible by their donation. The Boehringer Ingelheim Foundation funds our personnel, our training would not be possible without them. And last but not least, it's really also the imaging companies like Zeiss and Thermo Fisher, who really committed from the beginning with a lot of in-kind contribution. So I think this was really a joint effort to make it going.
I mean, imaging technology and microscopy is so useful across a wide range of biology that, and throughout Europe and everywhere else. I mean, I think virtually every scientist in the life sciences can appreciate that. And so the ability to train users in the latest technology is an incredibly valuable catalytic addition to life sciences in Europe that EMBL is providing. And I think we're very grateful for you doing that. I wonder if you could tell us a little bit more about what you're doing to help train people in these technologies.
Yeah, so here we really want to build on, or are building on, what EMBL is doing already in many different initiatives, but with a particular focus on other facility staff from across Europe, from the national facilities, so that we also try, once we can offer a new technology as a service here, that we try to push out the knowledge as quickly as possible also to the wider facility community and core staff that supports other researchers, so that ultimately new workflows can be implemented very quickly also elsewhere. Because independent of how many microscopes or people we have here, we cannot help everyone anyway. So this is the best way to spread the knowledge and get it out there.
And are there any lessons you feel you've learned from this journey in building the imaging center and setting it up that you could share with people wanting to carry out similar, not just in imaging, but in other facility situations in other places?
Yeah, I would say, especially from the project planning and from the construction side, I would say that if you have a chance, one should really think about a dedicated building, because also, as when I was still actively pipetting and doing imaging as a researcher, you always had to compromise on where you place your instrument. And then many of those highly advanced instruments cannot perform to the best of their ability if the building or the room does not allow it. So if you have a chance, that would be surely something to think about. So the integration with the research helps really to keep to provide the latest imaging technology. So you are really evolving with the research going on and with new workflows or new technologies elsewhere.
So the technology for imaging, of course, relies upon rather sophisticated instruments that are essentially provided to the research community by commercial companies, and more than one. And you mentioned the relationship that you build with commercial companies, but of course, to some extent, these companies also have some rivalries with each other. I wonder if you could share with us whether that introduced any special challenges about interacting fairly with as many of them as possible, or how you manage that.
Yeah, I think the key component is actually a relationship of trust. And here again, we are not, coming out of nowhere. This is built on EMBL Foundation. There's a long, successful history in relationship with companies, and this has built up this relationship of trust, so that also the companies think that if there is one place where you can work with several companies in parallel, then it could be EMBL. And then you have to make sure, of course, that you do not work on exactly the same type of technology with several companies in parallel, but that you separate also projects and respect their confidentiality. But at the same time, you try to openly share ideas and get them quicker into those companies for potentially having also quicker commercially available instrumentation out there.
Okay, I'd like to bring in Matthias. Matthias, I saw you raised your hand briefly, but perhaps you'd like to comment on this.
No, just a very quick addition. When you asked Steffi about, advice or clues for such projects, from the fundraising viewpoint, because when I'm in conversations with others, I'm not talking about EMBL, I'm just talking about the outside. Oftentimes here is that in raising for getting the money for such projects, it's clear that typically one needs the public side, so not philanthropy, but ministries, etc., will have to carry the lion's share of the cost. And the tendency is to find money first from them and then hope to fill the gap with philanthropy. And, you know, I don't think that that's the best practice. Quite to the contrary, I think to first try to find a philanthropic start that is significant, but at the end only a minor portion of the total cost, it helps in two ways. First of all, the philanthropists will have the feeling that their contribution has really a big catalytic effect, because, you know, they put in X, say, somewhere around 10 to 20%, and that immediately pays 80 to 90% dividends in that sense. And at the same time, the public side finds it much harder to reject when substantial philanthropic money is already on the table. That was all I wanted to say.
Okay. I mean, the funding of science is unfortunately key to being able to progress things. All I can say, Matthias, is I bought a lottery ticket today, and if I win, I guarantee I'll make a donation to the future of EMBL's development of service provisions as a philanthropist. So you have it's on camera, so I won't be able to get out of this. It does depend on me winning the lottery, but if I do, there you are. There's my promise. I'm so full of admiration for what EMBL does, because this is such an important, valuable service for all of European science. I really want to congratulate you, and I hope that EMBL gets, I don't feel it gets as much recognition as it deserves for the really important role that it does in training. And as well as training active scientists, of course, the other way that you help is by informing and stimulating interest in science in the next generations coming through. And I believe the Imaging Centre has also been a great way of helping to further that. And not everyone, of course, is able to come and visit the facility in Heidelberg directly, but many other people could also benefit from being able to see online the resources it provides and sharing that way. Maybe that's something, Stephanie, you could also mention as to how you see the importance of informing the public about the value of imaging.
Yeah, indeed. So I think the value is very high because imaging by its nature can visualize what science can do, right? And I think there it has a particular chance to visualize what we can do. And from the very beginning, we tried to implement this when the building was designed by having windows that allow access into the labs without necessarily going there. Also here, EMBL started very early to think on, okay, how can we get the public better engaged with us, with our science, beyond what is online available, for example, and also, especially locally in Heidelberg, how can we attract people to visit us even more? And this was when the idea about the exhibition came, with a new building, with having a little bit of space, which is usually a problem at EMBL. How can we merge maybe both ideas and get another dimension into this initiative? And this is where the world of molecular biology came into the whole building with us.
Yeah,
okay,
so
thanks, Stephanie. And back now to Thomas.
Yeah, so actually I'm very excited about this part, because I had been visiting a couple of times the exhibition, and I think is great that the exhibition is in the same site of the Imaging Center. I mean, so here to talk about that, we also have Agnes Szmolenszky, who has developed the museum part. And hi, Agnes, thank you for being with us.
Hi, it's very nice to see you all. It's great to be part of this discussion.
So as Stephanie was saying, you know, intent was to even more connect other parts of the society to the science. But I think what I want to ask you first is, you know, how did you go about doing this with an exhibition? This is something that the EMBL has never done before, or at least not to such a scale. And certainly, I can imagine that the expertise to set up a museal exhibition is actually, you know, not something that you had in every place at the EMBL. So what is the development of that? How did you manage to set up the whole exhibition?
Yeah, thank you. I mean, that was this question, and kind of answering this question definitely was the you know the foundation for success, and definitely even thinking about having such an exhibition, I think was an amazingly, you know, blue sky idea, which was very much initiated by Matthias, and who, but Matthias can correct me if I'm wrong, got Ian very quickly, I think, on board, kind of with the idea. And Jan Ellenbein has been very supportive from the very beginning, and of course more and more people at EMBL, but it has been a very, you know, it was something very, very new. The whole idea of sharing science and such a cutting edge science with the public was something that first a lot of people needed to understand, right? How is that possible? Why would that be good for people to understand? Can we adopt content in a way that they can really understand it, right? So there were a lot of questions around it, whether that's possible at all, whether it's not going to oversimplify it, right? What will, you know, visiting scientists say at EMBL who go to the exhibition, would it make, you know, would it be embarrassing for EMBL to have such a content, right? So if you think about all these questions, I mean, there were a lot of discussions about it, and even, you know, as we were getting closer to the opening, we still had discussions about it. And it was, you know, a big group of people. The project was managed at communications at the time at EMBL, several working groups focusing on different areas. And the big task was actually to select the research topics that we, you know, wanted to show in the exhibition, and then structure them in a way that can then organize the content that we have in the exhibition. Where the exhibition is very, very special compared to other museums or science centers, I think across Europe for sure, is that we really feature absolutely hardcore science and research projects. So it's a very much research-based exhibition, really featuring a lot of results coming from EMBL, a lot of scientists working or who worked at EMBL, the collaborative nature of science, a lot of different technologies, absolutely recent technologies are being introduced. So this kind of translation process was something very, very special. We were, of course, working together with external experts, right, whom we needed because EMBL is not an expert, you know, in the museum world. So we definitely needed them to translate content into language, into digital content that we can then engage our audiences with. From the very beginning, the two audiences we had on our minds was the public, right? Public meaning people without scientific training, and young people meaning those above the age of 14. And that again makes us special, because this is why the content can be so extremely research-based. We are not here to serve families and small children. But there again, if you talk to at the science centers, that is actually a value, right, that we add, because the usual science center would cater for families, which is of course very nice and very important. But then again, they cannot share certain level of knowledge that we can. So we really have a very unique position in this science center landscape now in Europe. And people love it.
Yeah, no, I loved it. I mean, I you know, I was struck by getting in the space. The building is beautiful. You have this giant LED screen that has, you know, beautiful images to a scale that just makes you go, you know, wow. And, you know, again, a lot of Drosophila images. So I'm very happy. So I wanted to ask you, I mean, did you take inspiration from museums that are existing, or from other, exhibition of this kind, or was it, mostly developing internally? And I mean, before you answer, I mean, I see that Nush is putting in the chat the images, but also the link of the virtual tour of the museum, which is really beautiful because you can, even if you're not in Heidelberg, step in and see. So did you get some sort of inspiration for this?
So we definitely, a couple of our colleagues, because at the time the development was not yet with my kind of team, but with comms, but they visited a couple of science centers in Europe for sure. We were, discussing how they feature, of course, different topic areas. But all in all, everything that you can see at EMBL is uniquely designed for EMBL. The content, everything is absolutely uniquely produced for EMBL. So in that sense, we are very special. And one of the kind of most special features what we have is that almost the complete exhibition and the interactives are digital. Mm-hmm. So if you go to any other museum, this is something, you could be very careful with, or wouldn't really add, not just because it makes it quite expensive, but also because it's quite challenging to maintain. However, we went for this very special kind of approach to be able to update content in a robust way, right? Mm-hmm. Because as EMBL, we wanted to make sure that we can keep this kind of cutting edge content, and we can update, research topics and projects. And that's, keeps us busy for sure, but this is really, really great. I'm grateful for visitors as well.
Yeah, and I think, for me, the mixing of science and art, so the beautiful display of things is one of the things that makes me even more relate. I mean, you feel it even more in your, in your heart and in your belly when you visit something beautiful that is also, eye-opening. So you were mentioning the fact that this was focused on 14 years old and over to make sure that you could develop the most updated concepts. But is it the only, type of visitors that you have? I mean, is it possible for people that are, for instance, come to a course at EMBL to visit the museum, or maybe, to perhaps have some of the activities of the courses and the people visiting the museum happening into the exhibition space?
Yes, so we are very, very happy to see that that course participants, so scientists coming to EMBL, and conference participants as well, are very interested to visit the exhibition, and they are themselves, although, right, they were concerned, and they are, very, well-known scientists, they absolutely enjoy it as well. They are very much impressed, and we get a lot of good feedback from them. And there again, I think EMBL is setting an example, right? Because we do get the feedback, them seeing it, that, this is maybe something we could, try at home, in our home institutions too. And wow, this is really working. This is not killing science, right? It's rather really, making science accessible. So I think that, again, we are spreading really the right message and inspiring the community, the scientific community as well, to share their science. And we do have a lot of kind of stakeholder visits to the exhibition as well. So, politicians, policymakers, all sorts of other, philanthropic, people, potential kind of philanthropists to EMBL come to the exhibition. And we do support, a selection of events as well happening in the space or around the space.
Mm-hmm. So I love the choice of putting it, in the same site of the imaging center, but I imagine that this is also quite a challenge, because you will have a lot of people, coming in with samples that have to use machine and instruments. So how did you manage the organization of these?
Yeah, I mean, that we were very, very lucky, because Jan Ellenbeck has always been very supportive. And then, of course, Steffi as well, but, we've definitely the first Jan's sign off, right? He has been extremely supportive, and we, discussed all the details at the beginning, and we said we start small scale, so that we will offer, and we still only offer guided tours to the exhibition. So we make sure, right, that people coming in are supervised in a way, but this again gives people a wonderful opportunity to get the most out of the exhibition. They get a 45-minute guided tour, and then a time on their own to explore it with our facilitation. And the same, of course, for the schools as well. They come only with organized guided tours and the workshops as well. And I must say that staff at IC has been really open and supportive too. We keep in touch on a regular basis. You know, we always tell them, you know, let us know if, you know, something is not working or in any way, you know, we are a bit disturbing your work. Of course, we learned a couple of things. Now we know which doors to close. if we have the visitors there, right? we came to put up signs that the visitor understand that, you know, their doors, they shouldn't open. But all in all, I think, you know, just entering that building is so impressive for people that they extremely, you know, respect everything and, you know, terms and conditions of being there, and really, you know, never ever had any issue with anybody. But, you know, we are always there. You know, some colleagues are always there, you know, looking after them and making sure that everything is safe and very enjoyable.
But I'm kind of curious, because I've noticed by, you know, walking in the building that, you know, you can see the actual lab and the actual machine of the imaging center from the glass, you know, walls. So does it happen that you have people visiting that want to know more, not about the exhibition, but what is actually happening there? Do they ask a question? What is that machine? What are the scientists doing?
Absolutely. I mean, if we could, you know, they would be, you know, thrilled, right, to actually enter the machine room, so to say. I mean, sometimes we do have some combined visit with Steffi, right, where we have both the exhibition tour and then we can actually visit some of the microscopes as well. Some of those which you can see through the glass. They would love to, for sure. And when we are showing the electron microscope grids, I mean, that's like the absolute, you know, highlight of, you know, at least seeing a little piece, you know, that goes into the machine. Yeah, it's extremely appreciated, and it's always a wow effect to see the technology.
So thank you, Agnes. This was you know, such an inspiring you know, discussion. And I mean, I think this is the best example to stimulate people to get into what, you know, the European Union calls citizen science, right? You know, get the citizens to contribute to science. So I really look forward to see what, you know, how this develops in the future, and whether there would be even more activities connected to the exhibition. I know, you know, it's called exhibition, but it really looks and feels to me like a museum, and I think every institute should have a museum. So I'll give it back to Angus so that we can move to the next step.
Thank you very much, Thomas. Indeed, educating and inspiring young people and the general population about the importance of science and the progress that's being made is another really fantastic thing that EMBL has been doing. And that was really great, Agnes, hearing about the steps being taken in this direction. And something else I think we can say about this this last year, as Thomas and I have undertaken this review of EMBL over the 5 decades in its 50th anniversary, is just how much science has progressed, and talking with people about their experiences and how they actually carried out science. You know, 5 decades ago when EMBL was first started, compared to how it's taking place just now, has really been a remarkable journey, and very eye-opening to remember the progress that's been made. And I'm very excited now to turn to our final guest today, Eileen Furlong. Hi, Eileen. And when I was lucky enough to work in EMBL Heidelberg back in the 80s and 90s, I worked in the gene expression program, and that's now Science Marches On, and that has metamorphosed into the genome biology program. And it's great to have you here as the leader of the program, Eileen, and to share with us, you know, your insights into how the whole study of gene expression has changed and how technology has contributed to that.
Great. Well, thank you, Angus. And yeah, always sorry that we never overlapped actually in your time at EMBL. And I'm sorry that I don't have a Christmas hat or something on. I tried to find one in the lab, but actually there's not. Yeah, exactly. Yeah, no, I'm very glad to be here to answer any questions. I mean, I think the study of gene regulation has changed dramatically. I mean, the unit is, of course, broader than that. I would say we look at all molecular processes. genes, but also RNA, protein, metabolites, but really at the large scale, and how they interact and cross-regulate each other, and how they change in different perturbations. Part of that is during, transitions, normal transitions like development, I do that, but also during disease states. So we have quite a number of group leaders also working in different diseases as well. But yeah, the field has changed dramatically. Actually, it's quite good timing, because thanks to the 50th anniversary, EMBL was writing a number of us got together. We were like, there was an idea to write a number of different documents, a little bit looking back at the history. And one of those is on sort of genomics, genome, including like genome archives, etc., which I think EMBL just published now. I just saw something on LinkedIn, actually about 5 minutes ago. Yes.
So I like to say something when I'm giving lectures, especially to some of the undergraduates when we're teaching, that I think you could reasonably argue that the biggest biological discovery and insight from the previous century was Francis Crick's central dogma that DNA makes RNA makes protein. And then I like to challenge the students by saying, I actually think in our modern era, we have to update that. Not that I'm making myself equivalent to Francis Crick, but just say that I think now, instead of DNA makes RNA makes protein, we can really talk about genomes make transcriptomes make proteomes, and then really deal with systems and really getting into biology as a system. And of course, James already earlier was talking about the really important fact that emergent properties arise when you consider interactions between complex components of systems. And so it's really exciting that you're able to go to the single cell level, but also measure like entire genomes and see this develop.
Yeah, I mean, I think single cell genomics has made a huge impact, actually. I think, it's percolating now to all levels of biology, but especially developmental biology. I think study of disease, also in evolutionary biology. If you think of, now it's, well, let's say single cell RNA-seq, it's probably been around for maybe 10 years or so. But at the beginning, we were all thinking of, identifying new cell types, identifying heterogeneity, but it's way more powerful than that. So now, I mean, oftentimes it almost negates some, necessity to do fact sorting. Like in my lab, we just, we do what we call brute force. So this is with homemade protocols for people out there. I'm happy to discuss these protocols. You can't afford to do this with 10x, but we take whole embryos and you can just pop them open, profile everything. So some experiments, we've done almost 1 million cells, and then you can see all of the cell types, and you can see that through developmental time, you can see all of the transitions, etc. And what it allows you to do is then phenotype things in a new way. So we can take mutants now and just de novo map those, like do the same experiment with the mutant, but map it onto a wild type time course. And then you can tell exactly what cell type, what stage of embryogenesis, what tissue, it's going to have an effect in. So it's extremely powerful, but of course the data is molecular as well. So you at the same time get all of this molecular information, which genes are changing. We're interested in regulation, so we look upstream at chromatin signatures, we get the enhancers, etc. So it's just, yeah, that is one area that's incredibly powerful. But there's also new developments. We have one group leader in the unit who's sort of pioneering single molecule genomics, which is quite exciting. So there it's orthogonal, it's to think about and compare to single cell. So you can really look at now which, we look at things like, for example, with chip, etc., chromatin IP, you think factors are co-localizing, and then you're making models that these two are working cooperatively, etc., etc. But if you have single molecule information, you can really see which ones are really co-localizing, which are anti, and then you can start to make occupancy models about those. And so this is actually a very interesting new development as well.
So when I was at EMBL, I still remember the excitement of the first ability to actually synthesize pieces of DNA or RNA and how that opened up things. And I want to give a shout out, I hope he's still online, but Brian Sproat, my dear friend, who was also working at EMBL at that time, he brought this technology, I believe, to EMBL and created the first ability on campus in Heidelberg to actually synthesize nucleic acids. But that's, if Brian's there,
Yes.
So Brian is, yeah.
Indeed. I've been doing this for, I'm retired since more or less 8 years now, but I still consult in this field for pharma and biotech, and it's gone a long way since making little bits of DNA. While I was still in MRC Cambridge, I did a gene synthesis for a human growth factor, and moved to the EMBL then and spent 10 years there. But then I learned to apply my chemical knowledge to tackling problems with biologists, and that's where I got involved with Angus's group and Matthias and Ian Matai and a lot of other people. And I think, this was 10 very exciting years to go and look. Let's make analogues of DNA and RNA, which are chemically stable, and we can use them to do all sorts of fancy experiments, which were imaging, microscopy, all sorts of fancy things, depletion experiments. I then went on later to develop ribosomes, or try to develop hammerhead ribosomes as therapeutics. I mean, we got somewhere. We did get an in vivo system which worked, although it always failed as a therapeutic, but we proved something, and that started off as a collaboration with Angus's lab, supported by Leonard Phillipson, who was very excited about that. We patented it. We got patents in the US, everything. And I went on further to work on aptamer projects, and now I'm involved with a few biotech companies who've got microRNA inhibitors going into clinical phase 2. So this is a very exciting time.
Very exciting. So Eileen, that's something that you see the whole history from Brian and the first synthesis to now, where it's become so powerful. We just don't even think about it. Young students, you just synthesize, and you can do it at scale and do remarkable experiments. How are you using this genome engineering and DNA synthesis technology these days?
Yeah, so great to meet you, Brian, because we haven't met in person before. Yeah, so no, it's actually incredible the developments in this, from in vitro assembly of very large backs. So we're actually making synthetic backs, so regions of 100 kb or so, to make, in our case, as I said, we're interested in gene regulation, but it's actually very complex if you look at endogenous loci. So many of us for many years have done the classic, let's say, genetics, where you're deleting things, but then you keep finding other things and you find a lot of redundancy, etc. So it's quite difficult. So now we're taking sort of more the synthetic approach. So it's a little bit, you heard from James, with kind of synthetic, I want to say embryos, but organoids. Also at the molecular scale, then the idea is to really build up complex landscapes. So then you can, know exactly what you're putting in there. All of the DNA is synthesized. You can stably integrate that, which is still a challenge, but stably integrate these large fragments into the genome. And then look at many different iterations and perturbations of that. So that's how we're using it. But like you said, it's very exciting. I mean, you can synthesize, there's a whole branch of people who are trying to synthesize then whole chromosomes. I mean, of course, people want to make a synthetic cell. People have done this to an extent in yeast, but the difficulty is then how to chromatize it to, but it's still, it's coming, I think. It's quite exciting.
So now that we're coming towards the end of this 50th anniversary year and our survey of EMBL across the decades, I think a great way to conclude is by looking to the future. And so Eileen, I want to put you on the spot. And now it's hard to believe that in every area of biology, including genome biology and genetics, AI and computational methods are not going to play a big role. But how do you see the next decade? If we're imagining Coffee with EMBL in 10 years' time, what are we talking about the big achievements of the next 10 years?
Well, I think, in my field, AI is already playing quite a big role. So, and I think actually genome regulation is one of the areas which is quite well primed to make an impact here. So, for example, we can now, the field, from DNA sequence predict transcription factor occupancy based on sequence very well, I would say, or at least pretty well. Predicting gene expression is a bit harder. But what we can't do well is to predict the effects of genetic variation. And that's kind of the holy grail. What you'd like to be able to do is look at your sequence and say, okay, this is going to affect now this transcription factor binding or this gene's expression. And that in turn would have an effect if we knew what enhancer it was at this stage of your life or in this condition, this tissue, and so on. And that is still very difficult, partially because we don't have the large enough training sets. And actually, this is where the synthetic DNA type of experiments, some people are trying to scale that up to try to learn from it. You know, the more data you can learn without evolutionary selection, the better in a way, some people would argue. So that's one area where I think looking back, we should have models where we can predict pretty well from gene expression. The other area is single cell genomics has really allowed us to build, they're still not that accurate, but gene regulatory networks at a global scale. And the idea there would be, you know, at least for me, one of my dreams would be to be able to predict what is the cell going to do in the next time point. You know, if you imagine now you have cells along in a trajectory, what branch is it going to take, or what effect with this perturbation would it take? So to be able to do that from the sequence, from the perturbation. So you say, okay, now you perturb this transcription factor, you have this deletion, etc. I mean, I hope, or I would imagine in 10 years' time, if we look back, I mean, imagine if we knew the sort of phenotypic space, as it were, and to be able to then have a model to tell us actually what perturbation or what mutation would be the best one to make, to turn it around in a way. Instead of us now, we're making it based on our own tuition and saying, okay, and then we're trying to explain the data and we're trying to train based on that data and so on. But rather to be able to say, okay, now if the model could predict the whole perturbation space, we had a feeling of that, like let's say if you thought of like yeast, a fitness landscape or something, and then the model would tell you, actually, no, don't be working over here, perturb over here. And this is going to be now the, you know, so something like that I can imagine, which would be very powerful in principle.
I don't know if Eric Cassanti is still online, but I also think a big shout out to Eric and everything that came from the Tara Oceans project, because that was a truly inspirational synthesis then of you know, information about like all the genes in living organisms in a whole area of the planet.
Yes. Well, you know, in the line of what was just said by Eileen, I think that there's still a lot to do with this data using artificial intelligence, you know, to try to look at the, I don't know yet how to do this because this is not too much my field, but yeah, I really think that there are things to look at because we have whole genomes now of different organisms, you know, that can be reconstructed from the metagenomic data. And there is probably a lot of things to do which are very exciting. I'm going to meet someone in Paris after the holidays who is working with artificial intelligence in genomics in Paris to discuss this issue to see what can be done actually. No, I think it's very exciting. Yeah, and you know, Tara Oceans is just the beginning because there are so many other samples that are going to be analyzed from other expeditions from Tara, but maybe Trek, I don't know what they did exactly, but there is also a lot of data that are going to come out from this. I think it would be interesting to see how we could link, you know, these field expedition experiments with in lab the kind of things you are doing in the lab to study the genomes and how they evolve or how they develop during development. Yes, I think it's very exciting to combine.
That would be great to kind of close the loop, but also, you know, I was thinking a lot of those organisms, the genes, I mean, are completely new and we've no clue what they do.
Exactly, yeah.
On the 3D level, maybe with AlphaFold predictions, we can start to have, you know, and complex predictions and so on. So some kind of transfer learning from different, but I totally agree. There's so much data and actually new ones, like Trek was inspired by your, you know, by Tara Oceans as well. So it's a similar, like more and more data will come from genes that we have no idea of what their function is, which is going to be
important for AlphaFold also because the more sequences you, independently of the structure of the 3D structures which we have through crystallography, the sequences are also very important for training AlphaFold. So I think I don't know if is there people who are working on at EMBL on the unknown genes of Tara for using AlphaFold or something like this? Or
I think there are some that are looking into it, but I don't know how systematically they're doing that. You should really ask Per Borg.
Yeah, I think I wish the problem with Per is that, you know, when I ask him questions, it's never very clear. He's also very often kind of fuzzy, so I never know exactly what he knows and what he does. But it's yeah, sure.
So if I can jump in, I think one aspect that I find very interesting that links, you know, what Eileen said and what Eric said is that, I mean, now with AI, we have the possibility of predicting the entire biological space, you know, of genomics or proteomics. And I was wondering for Eric, you know, because you have been so involved in finding new examples of genomes in all these out of the lab, real life organisms. How much do you think we still don't know of the biology by limiting ourselves to lab mostly organisms? And how much is there of that space that we are exploring with AI that is actually really existing, perhaps?
Yeah, well, I think we don't. When you look at the amount of new data we got and the unknown genes, I think we still don't know very much. The big problem, you know, in biology is that when we look at you know, organisms in the lab right now, we look at just a small window of evolution. You know, there has been so many different kinds of organisms, and we still don't know very much about them. So and even with AI, I'm not so sure how to address this question because it's so big. And it involves such big scales in terms of space and time. It's not easy to think about it, you know. So maybe AI will help.
But one of the problems I would think we might run into, though, is because, you know, when we're trying to, depending on what you're trying to train, what you're trying to find, but if you're trying to find function, like you said, Eric, we know we're in a very small part of the evolutionary space that
Right.
we've measured function. So the training is going to be very biased to one part.
Exactly. Yeah.
But yeah, I still think that, yeah, there should be hope out there. I mean, one thing to mention, sorry, Angus, but I was just going to say in terms of EMBL's contribution, just to put a plug in, I mean, it was because of actually a lot of the crystallography work that was done in Hamburg, Grenoble, the crystals were done there, but also very well curated in the databases at the EBI as well. That was the training ground that DeepMind could use, just to put a plug in.
Yeah, no, I think you do have to keep plugging this. As I was mentioning earlier, I think EMBL doesn't get enough credit for the
No, exactly. Yeah.
it has played. And, you know, even in a small way, that's what we are hoping to capture and help with in the whole alumni activities that we've been doing. And I wonder, as time's drawing to a close, Matthias, can I ask you to perhaps say a few words at the end or comment on how, you know, how you see EMBL contributing to the next 10 years and beyond of the future of science?
I mean, first of all, I agree with everything that has been said so far. I think what has been said so far sort of grows organically out of what we are currently involved in, and that includes a number of really exciting projects. But you know, what I like to add is, for me, the true spice in science comes from, you know, looking back over the past 10 years and seeing what on Earth one would never, ever have predicted when speaking about those 10 years one is talking about. And I just very, very sincerely hope that in the next 10 years, there will be all of these surprises. So, you know, when looking back, of course, 10 years ago, I don't know how many people would have predicted something like AlphaFold. You know, suddenly it was there. When I go to meetings of different types, what I see increasingly is, you know, biomolecules finding themselves completely in the wrong spots in cells or organisms, you know, creating big puzzles. And you know, surely there is something behind it. And for me, this is the spice. And for me, EMBL is the place where such out of mainstream fundamental science with a mechanistic angle should be pursued. And I very much hope that EMBL will have the opportunity and the guts to hire young people who want to take on such projects, so that in 10 years' time we look to surprises that are completely impossible to predict now. And that is at the same time my wish for EMBL.
Thank you. Thank you very much, Matthias.